Welcome to Dr. Glynis' Chicken Genetics videos. In this video, we'll look at gene variants and how they relate to chicken genetics. Genes for any particular characters may occur in slightly different forms called alleles. Alleles are alternative forms of a particular gene, and to understand genetics, you need to understand about alleles and how they work. Alleles come in pairs, one inherited from mum and one from dad. This is an example of the dominant white gene. It's given the symbol I, which stands for inhibitor of black. The white leghorns in the video have two copies of the dominant white allele and are said to be homozygous for that allele. The dominant white allele evolved by mutation of the wild-type allele. Chickens with two copies of the wild-type allele or two copies of the dominant white allele are said to be homozygous at the dominant white gene. Chickens like Safi are heterozygous at the dominant white gene. I can only tell this because I know she has a dominant allele from her mum and a wild-type allele from dad. Two other alleles at the dominant white gene are dun and smoky, and we'll discuss these in future videos. The different alleles of a gene occur by mutation of the wild-type alleles. Mutations change the genetic code, and there are various ways in which it can be done. For now, we'll use the analogy of changing words at random to understand a little more how mutations work. Substitution mutations change letters in the code. We can see straight away that sometimes the change makes sense and sometimes it doesn't. And even when it seems to make sense, sometimes it doesn't make so much sense when we look in context. A house may be green, but a horse and a mouse is hopefully not. The same can be seen for insertion and deletion mutations. Sometimes they make sense and sometimes not. Our government might be building a house or houses, but not a homes. But if we spell house just a little bit wrong, sometimes we might still be understood. There are also inversion mutations which swap half the code around so it's in the opposite orientation to before. And duplication mutations which duplicate a little bit of the code. As you can see, most mutations don't really make an awful lot of sense. Nature just leaves the ones that don't do too much damage to the organism they're in. Sometimes, though, animal breeders do maintain less favourable mutations, and we'll talk about this in some future videos. We'll take a quick look at our dominant white gene example to see how gene variants occur in chicken gene. An insertion mutation converted the original wild-type allele into the dominant white allele. A deletion mutation converted it into the dun allele. Both the dun and the dominant white inhibited black pigment. The dominant white allele itself was then mutated by a deletion mutation into the smoky allele, and black pigment was partly restored. Just to be clear, when we talk about original or wild-type alleles, we mean those that were first in the jungle fowl relatives from which chickens evolved. The alternative or mutant alleles make proteins that work the same way, or a little bit or a lot differently, to the wild-type proteins, or they may not work at all. Some important things to remember from this video are that alleles are alternative forms of a gene made by mutation and represented by a short code. Each gene is represented twice in any particular chicken. If the chicken has the same alleles for that gene, it's said to be homozygous, and if it has different ones, it's said to be heterozygous. Some alleles can cause deleterious effects, and we should do our best to try and select against those. Thank you very much for watching this video. If you have found it useful, please like, comment below, and share it with your friends or on social media. Subscribe so that you're notified of chicken genetics videos as they come out. I hope to see you very soon.